So we're right here just about to wrap up Chapter 8. We're talking about different ways to select mutants if you wanted to do that. And we talked last time about direct selection. Remember what direct selection is and how you can do that? When can you use direct selection? When you're sleepy.
Isn't it when you have something that you're really resistant to and put it in?
Almost the opposite, if I understood what you said. When you have something that kills the normal cells or stops their growth, and then you select for mutants that are able to grow in the presence of whatever that chemical is. So whenever you can add something that inhibits the growth of the normal cells, one way or another, then you can select for mutants so if you wanted to check look for caffeine-resistant mutants of E. coli, you could throw enough caffeine in that the normal cells couldn't grow, maybe died, and then you'd select for those that did grow in the presence of that excessive caffeine, and then you'd look to see if they were better students or not, or something like that. Yeah, something like that. And so that's direct selection. So the easy thing to imagine is antibiotics, because you know that some microbes are inherently susceptible to antibiotics. That's the Kirby-Bauer method, what you learned in that part of the laboratory. And so you could take one of those that's susceptible and then allow it to grow in the presence of the antibiotic. Only those that mutate, pick up some change in their genome, will be able to grow in the presence of that. That's what that question was about on that exercise. If you didn't know, when you had those two colonies that were growing down in the zone of inhibition, it was really asking you what might have happened. And there are two ways those could have arisen. If there was some contaminant in the cells that you put down there that were already resistant, that would give you those colonies. Or if those guys mutated and became resistant to the antibiotic, then that would be the second way that you could get those. The other way we can select for mutations is a little bit, in fact, a lot more complex. And if you can do direct selection, if you can figure out a strategy where you can use direct selection, that's the way you want to do it. Because the second way takes a lot more time, it's much more tedious, and if you want to screen the tens of thousands, hundreds of thousands, or even millions of cells that you need to be able to find a few mutants, then the second method is pretty laborious and takes time and energy. I think I talked to you last time. This is the direct selection procedure. So over here they put media containing streptomycin, nothing grew, and except for one colony. And over here on the right hand side there's media without. You can see that everybody grew. In that, on both of those plates, in that case you'd put literally 10,000, 100,000, maybe a million cells out on that plate, and most of them wouldn't be able to grow. So this indirect method is what you need to do, say if you're looking for an auxotroph. We talked last time about E. coli can make all 20 of those essential amino acids, but you might want to select mutants that can't make one. And last time we talked about tryptophan auxotrophs, their inability to make tryptophan. So if you wanted to select that particular auxotroph, there's no way to do it with direct selection. You'd have to use an approach like this. And in this approach, you plate out the organisms that you're looking at, and so that's what we have over here. That's called a master plate, and that master plate has all the bugs growing there. Hopefully in that mix of colonies that are growing there you've got the prototrophs, but hopefully you also have some, in this case, auxotroph, mutants that can't synthesize some amino acid or something on that plate. And that particular media has to be rich. It has to be able to support the growth of not only the wild types but also the mutants. And so everybody will hopefully grow on that particular plate. And then once you've got them growing on that plate, you can use this setup right here, and all this is either a wooden block or sometimes a metal block that's shaped just the right diameter to fit down inside of a petri dish. But before you put the petri dish over it, you put a piece of sterile velvet. The ladies in the room probably know what velvet is, but guys might not. It's a pretty thick plush fabric, but if you look at it closely, what you see thousands and thousands of little fabric fibers sticking upward from the main mat of the fabric. And you can sterilize that in the autoclave, and you can then put it over this metal block, and essentially what you have is tens of thousands of inoculating needles sticking up from this surface. So then you take your master plate, this guy, and press it down onto that velvet, and some of the bacteria on those colonies will transfer onto the fibers of the velvet. And then you pull that plate off, you can put the lid back on, you can save it, and then you've got those colonies transferred here onto the velvet, and then you can do the next step. You can go in with a couple of different types of media. One that you would probably use is this glucose salts media. Remember, that's a minimal media that auxotrophs wouldn't be able to grow on, because auxotrophs need some additional growth factors, some additional nutrition, and they can't grow on there. And so that would be the plate that you're actually selecting for mutants that can't grow, or to see those mutants. And then you'll also want to do this plate, which is the same media as we got over here. So these two are both the same, and this one's kind of the control on the left. Everything should grow on this plate. The mutant should grow. The mutants are the auxotrophs, and the prototrophs should be able to grow on that plate as well. And so what you're doing then after you incubate those is you look at those plates and you see who grew on those plates, and you should see all the same colonies on this plate that's on the left as you did on the original plate. If everything went well on the transfer, you would see all the same colonies represented there. And then you start to examine this minimal media plate. And on that plate, because the prototroph can grow there, you should see all the colonies of the prototroph, the original wild-type cells represented there. Did I pass a roll sheet? Okay, good deal. So you should see the prototrophs growing there, but you'll see that in some places there'll be a colony missing. Some colonies won't grow. Those are presumably mutants that can't make something that the original prototroph could make. And then you'll either go back to this position on this replica plate, or you'll go back to the same position on the original plate, and you can pick the colonies from there and start characterizing those colonies. An easy way to do it, if you're looking for amino acid auxotrophs, some that can't make an amino acid, is first throw in all 20 essential amino acids into that minimal media. Supplement with all the amino acids and then plate back, and then you can see, yep, for sure they can't make one of the amino acids, because when we put it back into the media, now they're able to grow. And then you can start using smaller groups of 5 of the amino acids, and figure out which one of the amino acids it really needs. And then you can start looking closer at the genes that are mutated to see what's going on in that case. So this is replica plating. You can use it for a lot of different mutant selection procedures, and anything that is going to be auxotrophic, you can use this type of selection for, because the original media, this media, are all providing the nutrients that are needed, but this media right here is selective, and the mutants won't be able to grow. And then you can compare the two types of media and look to see which colonies are missing on this particular selective media in this case. So that's replica plating. There's another way. If you plate out spontaneous mutants on those replica plates, you'll end up plating out hundreds of thousands, millions and millions of colonies and replica plating in them, and you still might not find the mutation that you want, if that mutation is relatively rare. But there's a way to enrich for those mutants that are auxotrophic, and that's what this procedure is. And this procedure is called penicillin enrichment. And it's useful with bacteria, but you can think of similar strategies that would work with other types of organisms as well. And in this case, the wild-type organism has to be susceptible to penicillin. Penicillin has to be able to kill the wild-type organism. Do you remember the mode of action for penicillin? What does penicillin target? Cell wall. And what part? Does it break down the cell wall? Inhibits the synthesis of the cell wall, right? And so what kinds of cells are making cell wall? Rapidly dividing cells. They need to synthesize peptidoglycan so that they can get bigger and ultimately divide. So those growing cells are synthesizing peptidoglycan, and this procedure takes advantage of that. So we do this experiment, and we've got this tube, and down inside of this tube we've got both the prototrophs and the auxotrophs. Just the majority of the cells, the large majority of the cells, are going to be prototrophic. They're going to be wild type. And just a few of them are going to be mutants that are auxotrophic. So again, if you plate those out and do the replica plating, then the large majority of them are going to be prototrophs, and you're going to have to scan lots and lots and lots and lots of colonies to find those mutants that you want. But if you do this instead and take those cells and put them into media that has penicillin in it, assuming that the original prototroph is susceptible to penicillin, And this media is glucose salts media, so it's that minimal media where the auxotroph can't grow. Right? And so you put them in, the prototroph will grow in there fine, except for the penicillin kills the prototroph as it's trying to divide, because it blocks the synthesis of peptidoglycan. The cell wall gets weak, the cells die from the presence of penicillin. But those auxotrophic cells are just sitting there. They're not going to make peptidoglycan because they can't make amino acids. And so they're just sitting there. They're not growing. They're not synthesizing peptidoglycan because they're not enlarging their cell size because they can't make proteins. And so they just sit there and stare and wait, and the penicillin can't impact them. And so you can enrich for those auxotrophs. And if you plate it out then, now you have to plate back on a media that supports both types, and you plate out, and now you get a much higher proportion of the auxotrophic cells coming out because most of the prototrophic cells got killed by the penicillin as they were trying to grow back in this original environment again. Before you plate it out, you would do something like hit it with an enzyme called penicillinase, an enzyme that breaks down penicillin, where you could wash the cells to get rid of the penicillin and then plate them out on these plates. And when you then go to do your replica plating, you'll see a lot more auxotrophs in the colonies that grow proportionally to what you would have seen previously. Okay, so that's penicillin enrichment, and you can think of other types of enrichment strategies if you use other types of chemicals that would, again, kill the normal cells but not hit those auxotrophic cells. Okay. All right, so now I need to ask you guys a personal question, because some of you do look like you just woke up. And so I need to ask you, how many of you got in that box thing that has water that sprays over you this morning before you came to class? All right, some of you are brave and raising your hand. Some of you are just raising them because you don't want to not raise your hand. But that gives me an idea. Good. And then in that box thing, you usually have this bar or bottle of something that you rub all over your body, right? And you probably did that, right? And some of you got another bottle and you rubbed it on your hair, and then maybe you rinsed that out and got another bottle and rubbed it on your hair. And others of you, even if you didn't raise your hand to that first question, you got up and you put something under your arms. Why did you put all of those chemicals on your body? So you'd smell good? Yeah, I learned a new word in yesterday's class. One of the guys said, so I wouldn't be ashy. I didn't know that word. Do you guys know that word? All right. I didn't know that word, but now it's part of my active vocabulary. So you would smell good. And you use some words that I would understand, but now I know ashy. You can use ashy freely with me. I actually went to the Urban Dictionary last night to figure out exactly what it means. But seriously, you just rubbed all kinds of chemicals. Weren't you afraid that you might get cancer? Weren't you afraid that they might mutate your cells and that someday you would die?
But everything gives you cancer nowadays.
Everything gives you cancer nowadays, so you might as well not be ashy while you're getting cancer. Is that what you're thinking? Okay. Starbucks does?
Yeah.
This is the neighboring campus center coffee. It's been tested in California not to cause cancer. So.
How can we tell California?
It's not on the label anyway. So seriously, you didn't worry about putting those chemicals on your body. Some people were worried about that, though, and some people want to be more careful than you footloose and fancy-free college students appear to be. And so some people really don't want you to get cancer because it costs you and it costs society a lot of money. And so all those chemicals that are in those things that are applied to our body regularly have to be tested by the FDA, the Food and Drug Administration, to make sure that they don't cause cancer. And for lots and lots and lots of years, they took these really cute little fuzzy baby bunnies, and they shaved their backs on a daily or weekly basis, and they applied lots and lots of these chemicals that are in the shampoo and the soaps and the lotions and the deodorants that you're going to put regularly on your body. And they said, little bunny, we love you, and you're cuter with fuzz on your back, but here, test this chemical to see if it causes mutations and ultimately causes cancer. And those bunnies were genetically engineered so that they couldn't do some of the things that normal bunnies and you all can do, like repair damage to DNA. And so the mutations would show up faster in those bunnies. But if there was a high prevalence of bunnies that had mutations in that colony when they were exposed to a certain chemical, then it would be deemed probably not appropriate for use in human populations. And on the other hand, then that's part of the reason we're going to the next part, is that some people said that those cute little funny buzzies should not be subjected to having their back shaved and these potentially dangerous chemicals that actually make them smell good be rubbed onto their back. And so there was a lot of pressure to not use that approach. And so about 30, maybe 40 years ago now, a guy named Bruce Ames came up with this test that doesn't use cute little bunnies. And I keep getting the wrong pen. The Ames test doesn't use cute little bunnies. It uses salmonella. And it seems nobody complains when you kill salmonella cells. I don't know why, but nobody complains about that. They're actually nicer to work with than bunnies. If you've ever played with bunnies, one of my second job, I guess, was in a pharmaceutical chemistry lab, and we had to inject bunnies with all kinds of drugs to see how they just went throughout the body, to see if they made it to the brain, to see if they made it to the heart, to see if they made it to that place. And bunnies scream, and they're really not as cute and friendly as you might think they are. They bite too, yeah, if you don't have them subdued and good gloves on, and they squeal like pigs. But anyway, they're not fun to work with, and they're expensive too. And besides that, we had to keep them in cages for 6 months or a year, rubbing these compounds on them to see if they were going to develop cancer, and that's pretty expensive. And so bacteria, are much easier and cheaper to grow. And so this Ames test uses salmonella, and it uses specific strains of salmonella, and these strains are all requiring histidine. What's another way you could say that a bacterial strain requires histidine? Thinking of an A word. They're histidine oxytropes. Beautiful. They can't synthesize histidine anymore when they used to be able to. So they mutated these guys to not be able to synthesize histidine. Okay? They also selected strains that didn't have those DNA repair enzymes that we've talked about. So if they get any tweaking going on in the chemistry of their DNA, like DNA polymerase inserting those mistakes, then they're not going to be fixed. So there's no mismatch repair. There's no excision repair in these strains of salmonella. And that means that mutations happen at a much higher frequency anyway. Okay? Because there's none of those systems to repair those. And they selected a strain that also had one other characteristic. They selected a strain where the membranes, remember the salmonella is gram-negative, got two membranes, inner outer membrane. The membranes are much more permeable, so chemicals can get down through. They can diffuse into the cell more readily and get down inside. And so they use this strain, and then you can then use direct selection to see if these chemicals that you might want to rub on your body, or pretty much any chemical that's produced these days. Pharmaceutical companies have large banks of organic chemists that's all they do all day long is synthesize new organic compounds that haven't been made previously that potentially could have pharmaceutical effect for some disease in humans. And that's their job all day long, is make new chemicals that haven't been produced before, and then they'll give them to other people in the company and say, does this have any effect on these cancer cells, or does this have any effect on this type of situation? And before they can use those, they need to see if they're mutagenic. If they're going to cause mutations down in the body of a human, then that might be a concern. But when you rub those chemicals all over your body, besides not being ashy and smelling good, what else happened? Well, you wash off the bacteria, but I'm thinking more about the chemicals. Did the chemicals stay on your skin? You hoped so. I see head shaking, but in fact, most of them don't. Most chemicals absorb because just a little bit below the surface of the skin, there's capillaries, and some of that chemicals gets into your bloodstream. And when it gets into your bloodstream, it makes its way to your liver. And when it makes its way to your liver is there to detoxify chemicals that get into the bloodstream. But as it's doing that, sometimes it actually makes modified chemicals, breakdown products that are more toxic than the original chemical. And that's just part of the job of getting rid of all those chemicals. It happens naturally in the body. And so to make this test even more lifelike, they take livers from rats, and apparently you can take livers out of rats but not shave bunnies' backs. Take livers from rats, grind them up, extract all the enzymes that are in the liver, and then expose the chemicals to that rat liver extract before you actually test them. And so that's part of this test as well. And then you look to see if the chemicals are going to be mutagenic or not. Mutagenicity and carcinogenicity aren't necessarily always directly related, because remember, like the bunnies, these guys are set up to have cancer, if you can have cancer in bacterial cells. These guys are set up to have mutations because all of those DNA repair enzymes are knocked out. You guys must be proficient because you're still alive and you're breathing and you're not getting sunburn sitting here. And so you must have those DNA repair enzymes functional in your body. Plus you also have a functional immune system that's looking for those renegade cells that are heading off toward cancer and trying to kill those renegade cells that have gotten mutations. And so just because something is mutagenic doesn't mean that it's necessarily going to cause cancer. Depends on what happens down inside of the body. So that gives us a way to test for chemicals that are mutagenic. And the Ames test works something like this. In every test, you always have to run controls to make sure the system's working well. And so this left panel is what you would do in the control situation. And the first plate here, basically, this plate is glucose salts agar. Remember, glucose salts is that minimal media, and auxotrophs can't grow on that media because there aren't, in this case, amino acids in there. And we're working with a histidine auxotroph of salmonella. So you plate these cells, and they won't be able to grow because they can't make histidine, and there's no histidine in the media. You plate them on that first plate, and then you might add the diluent that you're going to put your suspected mutagen, or at least the chemical you're wanting to test, on those cells as well to simulate the situation. And then you incubate those, and then you look at the plates to see what happened on those plates. If everything works normally in this control situation, you shouldn't see very much growth. Even though you might have put 100,000 or 1,000,000 cells on that plate, you should see very few colonies, but you will see some. And those guys are revertants. Those guys are cells where when DNA polymerase was copying the histidine gene, it made a mistake that fixed the original mutation that made it a his auxotroph. Okay? So DNA polymerase makes mistakes, you know that, and DNA polymerase is always making mistakes, and occasionally it'll make the right mistake to fix the mutation that was introduced previously. And those guys we call revertants, and you're going to see those on the control plates, just a few of them, because that's happening. And whenever, remember, DNA polymerase is making mistakes, the repair enzymes aren't there, so the mistakes are accumulated, and some of the cells are going to go back to His plus. Okay? On the bacteriophage are going to come and infect this first cell. The first host cell becomes infected, and many bacteriophage, one of the things they do when they get down inside of the cell is they make enzymes that start chopping up the chromosome of the host cell. If you chop up the chromosome of the host cell, then the host cell pretty much can't divide anymore, right? Now you've got a nice comfy place to live, grow, have babies, and do the rest of your life cycle, and that's what goes on here. The bacteriophage then, they break up the chromosome, and then they start expressing all their own proteins. They hijack the ribosomes from the cell to make their own proteins, and they make their own genome. And once they've made their genome, they start packaging their genome down inside of those virus particles. But if you look closely at this picture, and in reality it's hard to see, let me just do this for just a second. One of those phages, instead of getting viral genome, got part of the host chromosome put into the virus particle. So now we've got a virus that can go and attach to another cell and inject DNA into the other cell, but the DNA that it's carrying is not viral DNA. And that's how the next part of the process plays out. When that phage then goes and finds another host cell, oops, I didn't quite get to the next page yet. When that phage finds the next host cell, it'll attach to the host cell. It'll inject the DNA that it's carrying, but this time it's not viral DNA. This time it's a segment of the genome from that previous host. Again, it might be the same species, it might be the same strain, but it might be a different strain or species. And if it is, it's bringing in DNA that wasn't in this second host that the first host had. And as that happens, the DNA will get transferred, and the DNA then potentially will become part of the chromosome. If it becomes part of the chromosome and it was different from what was there previously, in other words, if it was a different strain or a different species, then now the progeny are different. Some new genetic characteristic got moved to this second host because the original DNA that was there is being replaced by this DNA that's being transferred. So you probably noticed that there are a bunch of video links on each one of these pages. I put those there because I think some of you have forgotten about all the resources that are in the Nester website. If you go to the PDF and just click those links, it'll take you to the page for this chapter, and there's lots of great videos that are going to explain these processes in a little bit more detail. I would encourage you to do that. I think this chapter has 15 or 18 videos that go along with these concepts that we're talking about. So go click those links in the PDF that you have access to through my campus, and you'll be able to watch those videos, and it'll make more sense of what's going on in these processes for you. The third process is called conjugation. And there probably should be a black box right over this area, but basically these two bacteria are having sex, and they're doing this process of conjugation. They set up this, the polite way to say it is an F pilus. The other way that it's said is a sex pilus that attaches and connects the two bacteria together. And basically it's a tube, and it appears that the DNA is pushed through that tube from whichever is the donor cell to the recipient cell. In this particular picture, it's hard to tell, but if we say this is the donor cell, the DNA from this guy would be pushed through that tube, and ultimately the DNA would make its way over to this second cell in that picture. So this is conjugation, and one way that it was used to demonstrate that the DNA is actually going through that F pilus is that you can add an enzyme that's called DNase. And DNase will degrade DNA. And so if the DNA was somehow spiraling around the outside till it got over here, DNase would degrade it, and then the DNA theoretically wouldn't make its way across. But apparently the sex pilus protects the DNA. There's still some people that think that the DNA goes on the outside of the sex pilus and that the DNase is just not able to get to it when it's going outside. And that's one of the beauties of science. You get lots of debate and lots of experiments to try to figure out which way is right. We would still be thinking spontaneous generation happened if some people, maybe we still do, but if people hadn't done those experiments and figured out that it was the endospores that were causing what was being seen in some of the experimentation. So sometimes in cells there are plasmids that we call conjugative plasmids. In fact, most of the time that we see this conjugation happen, there's a plasmid that codes for the proteins that make up this sex pilus. So if the cell doesn't have that plasmid, it doesn't have the genes to make the sex plasmid. Okay? The F plasmid, we call it. And so usually those plasmids persist as replicons, because remember, a replicon is a piece of DNA that has an origin of replication that can replicate and persist without integrating into the chromosome. And so usually the conjugative plasmids happen that way. And the very best studied of all of those is this plasmid that's called an F plasmid. And the F stands for fertility, because those cells that have the F plasmid are fertile. They can transfer DNA from their cells to other cells. Okay? And basically, a lot of studies have been done to look at that, but conjugation happens kind of like this. The cell over on the left here has the F plasmid, and therefore it can make the F pilus, because the proteins are coded for on that plasmid. And so it makes the pilus, and it makes that connection between another cell and the second cell. The recipient cell is called an F minus cell, because the F minus cell, the recipient, doesn't have the F plasmid yet. So once that conjugation pilus is set up, DNA starts to transfer. And remember, DNA is double-stranded, and an enzyme comes in and it nicks one strand of the DNA. And then that strand, DNA polymerase, comes in and DNA polymerase starts to replicate right here from this end, and as DNA polymerase moves along this way, it pushes that strand that is nicked, and it pushes it and makes it separate from the other strand as DNA polymerase continues to replicate along. And then this strand is pushed through the sex pilus as this circle is replicating. DNA polymerase will continue on along that circle, and as it's continuing on along that circle, you can imagine that more and more of that strand is going to be pushed off, and more of the strand will be pushed through the sex pilus as that replication process happens. And pretty soon, if there's no interruption, the whole plasmid will be pushed through. But if you think about the model I just tried to draw for you there, as one strand is being pushed through, the rest of the plasmid is being made double-stranded, right? So the plasmid is staying double-stranded, but one single strand is being peeled off and pushed through the sex pilus and transferred over. So when that strand gets over to the recipient cell, it has to be converted to double-stranded, and then it'll replicate as a plasmid down inside of that. And here's the question I'm going to ask God when we get to heaven. When bacteria have sex, they all become male. So it's one indication that, ladies, I'm sorry, but the male species is the dominant species of all things, right? Because in bacteria, everybody becomes male, and God wouldn't make a mistake. If it was wrong, God wouldn't do that, right? No, I'm just kidding. But in bacteria, that's the way it happens. As soon as the cell receives the F pilus, it converts from the F minus, or female, if we want to use that analogy, and then it goes to the F plus, or the male, and then it can conjugate with other cells. It can find other F minus cells and transfer that DNA to other F minus cells. There's a lot of text on this slide that I didn't talk to you about. But basically, the enzymes cut the DNA at this very specific place. It's called the origin of transfer, and then the recipient becomes F positive. There's another thing that happens sometimes with these plasmids. Sometimes they don't stay as plasmids, but sometimes they integrate into the chromosome. And if they become part of the chromosome and then they start to transfer, think about what could happen then. If these plasmids become part of the chromosome and then they start to transfer, they wouldn't just transfer plasmid genes, they would also transfer chromosomal genes. And they would transfer the chromosomal genes over to the recipient from that original host cell. And that's what we see happening here. And these guys are called HFR cells. It stands for high frequency recombination because they move parts of the chromosome, and what they saw was that you could get recombination between two strains by moving parts of the chromosome. In fact, they used this technique before we had molecular biology. They got these plasmids to integrate into different parts of the chromosome along the chromosome, and they had lots of different strains that had the integrants in hundreds of different places on the chromosome, and then they would start transferring. And you could let them have sex for 5 minutes, or let them conjugate for 10 minutes, or let them conjugate for 15 or 20 or 40 minutes, and all you have to do is vortex the tube to interrupt the conjugation process in bacteria, and then you could see which genes transferred based on the position of that plasmid in the chromosome. The ones that transferred the highest percentage were going to be the ones that are closest to that position where the plasmid integrated in the chromosome. If this gene transfers 100% of the time in 5 minutes, then it's really close to where the plasmid is. But if this gene only transfers 20% of the time in 5 minutes, but 90% of the time in 10 minutes, then you know it's a little bit further away from the place where the plasmid integrated. And that's how they mapped the genes on E. coli initially, was by integrating these plasmids into all the different positions on the chromosome and doing these experiments, and then figuring out, okay, if it integrates here, what's closest to it and what's further away from it on the chromosome. And they did lots and lots of experiments to figure out the order of those genes that they knew about and their position around the chromosome based on this type of mapping. So there's a lot of other cool stuff that you can do. Once the F plasmid forms, it can also jump back out. I mean, once it integrates into the chromosome, it can jump back out of the chromosome. And if it does that, it doesn't always jump back out exactly like transposons would. Sometimes it jumps out and it takes part of the chromosome with it. and sometimes it jumps out and it leaves part of the F plasmid in the chromosome. And when it does that, it becomes a defective F plasmid. It might still be able to transfer to another cell, but if it loses the origin of replication and it transfers, then it's not going to be able to survive. Or it might lose some of the other genes as it's transferring when it's that type of plasmid. And when it converts that way, it's lost part of its normal genome as far as the plasmid goes, and it's got something else. We call it an F prime cell. It's got the F plasmid. It can still make the sex pilus because the genes are there, but the F plasmid is modified, and when it gets transferred, it can't do everything. Many times those F prime cells, once they transfer, the recipient can't make the pilus because the genes for pilus production might not be on the part that's transferred over anymore. So that has a lot of utility as well, and lots of application to that. So the other thing that happens with those HFRs is they tend to pick up little segments of the chromosome, and you can actually see that happening here with segments of the chromosome. And then when you get transfer, you get lots of different pieces being transferred across and different bits of the genome being across. And that's another reason they're called those HFRs, high-frequency recombination. Transfer brings genes from lots of sites on the chromosome. It takes about 100 minutes for the full plasmid to conjugate over, to transfer over. So from this information, you're starting to see that genomes aren't nearly as stable as we thought they were. It's a little bit scary because we've got lots of bacteria out there that are resistant to the antibiotics that we have. And if they can transfer genes, and if the recipients can become resistant to those antibiotics, theoretically they could quickly win. And that's why genomics is important, because like I told you before, people are sequencing thousands, tens of thousands, even hundreds of thousands of different isolates, different strains of those organisms that potentially are pathogenic to us, to see what kind of characteristics they're picking up. And a year and a half ago, I went to a conference, and there was a guy that is just, every time he can grab an E. coli colony from any place, he'd be happy to collect material from you all if you gave it to him. He would isolate those E. coli strains, and he would sequence their entire genomes, and he'd sequence thousands and thousands and thousands of them. And what he finds out as he's doing this is that virtually no two isolates are alike. There really is no wild-type E. coli anymore. There are thousands of wild types E. coli out there now, and most of them have picked up DNA from other organisms. And some of them, we call them E. coli, but 70% of their chromosome is actually DNA that they picked up from other places. So when do you start saying it's not E. coli anymore? Just because it does metabolism and it looks like E. coli and smells like E. coli on plates, do you still call it E. coli even though it's mouse 70% Shigella or 30% Salmonella, 40% Shigella, 20% Staph aureus? When do you stop calling it E. coli anymore? And so that's a real big problem of microbiology right now. We've got all of these isolates, and biochemically they characterize as E. coli, but when you start looking at the other genome, some of them may have large portions of their genome that aren't. And so then we have to start characterizing what we call the core genome, the genome that every E. coli has, and say, okay, it's got these, and it's got these, and it's got these, so we're going to call it E. coli, but now it's a different strain, and it's picked up all these other bits of DNA along the way that aren't parts of the core genome, and that's the mobile gene pool. That's the DNA that's moving around in those communities and being transferred between organisms, and that's the part that's concerning. Most of the time, these guys are plasmids. Sometimes they're moved by transposons. We didn't really talk about those F plasmids, but let's say, for example, that bacterial cell had a transposon on the chromosome. And the transposon was, say there are two transposons on the chromosome, and the transposon jumps into that F plasmid, and it takes that piece of DNA from the chromosome onto the F plasmid. And then that F plasmid transfers that piece of DNA over to another species. Now that species again got a big chunk of that particular chromosome. So transposons play a big role, and these guys are really significant. We call them genomic islands. They're places in the chromosome that have DNA that's not from that particular organism. And we can see them, and they call them genomic islands because when you start sequencing them, organisms typically have a characteristic sequence. They'll have a certain number of GCs compared to ATs, and you'll see certain arrangements of bases. Even though we talked about all 61 of those codons working, some of them are going to be preferential. And you'll start to see that those preferential codons are used in the genes. And now we see other genes that are there that don't use those preferential codons. They have codons that look more like the codons that are preferred by a different species. And we can begin to identify those as genomic islands, pieces of DNA that have come from someplace else, landed in this chromosome, stabilized in this chromosome, and presumably giving that particular organism some other benefit. If you think about it, if they pick up DNA, there has to be some benefit. E. coli normally can replicate every 20 minutes, but if its genome just gets 25% bigger, that's going to slow down the process. And that takes more energy to replicate that much more genome. There has to be some natural selection benefit for that piece of DNA to be able to survive in that particular population. Your question?
Yes. If you had a transposon that went to the plasmid and then it was transferred to the other, does that mean that the original cell just lost the ability of whatever that transposon did?
Remember, most transposons can jump and many of them can duplicate and make copies of themselves, so it depends which way it moved. If it jumped out of that position and didn't jump someplace else back in the chromosome and just jumped to the plasmid, then it might have potentially lost that. But in reality, no, because if we're talking about these conjugated plasmids, the plasmid still has a copy, right? One strand gets over, but the plasmid still retains that copy of the transposon, if it moved that way, if it moved by conjugation. Okay, so we can begin to look then at the mobile gene pool, and as we've looked, in fact, probably most bacterial species, most archaea species, we can find plasmids down inside. Plasmids are pretty interesting, and we actually take advantage of them. Some yeast even have plasmids. Saccharomyces that you looked at, they're plasmids that can replicate down in Saccharomyces, and we make some artificial plasmids because we want to get bits of DNA down inside and get them to replicate down inside and carry genes over that we're interested in. Usually plasmids are double-stranded DNA, and usually they're circular, although there are some linear plasmids out there as well. They have to have that origin of replication. Generally, they're non-essential, but they obviously give the cell some ability. So if they're not essential, there are tricks that you can play to get the cells to lose those plasmids, because if they lose them, their cell's still viable. They just lost some traits. Maybe it was antibiotic resistance, and if there's no selection for the antibiotic, then you could select for strains that didn't have that plasmid carrying those genes. Usually there's just a few to maybe 1,000 genes. Some plasmids replicate in what we call low copy number. In other words, they maintain at 1 or 2 copies per cell, just like the chromosome would. But others replicate at high copy number, and they might have hundreds of copies in the cell at any given time. Those are the guys that when we do molecular biology, when we want to clone genes and we want to move genes in, usually we use the high copy ones, unless the protein is toxic to the cell, and then we might switch to a lower copy number to get less of that protein produced. Most of these plasmids have a relatively narrow host range, meaning they won't replicate in a lot of other hosts, but you can find some hosts where they can. And again, that goes back to DNA polymerase and all those other proteins that have to start the process of origin of replication. But some of them will replicate pretty broadly. They'll replicate in both gram-negatives and gram-positives, and so those are very useful for moving genes around. If we look at this slide real quick, you can see, because some of you are wondering, why is this guy talking about plasmids? They're just bits of DNA that replicate, most of them autonomously, most of them double-stranded and circular. Why is he talking about these guys? But I want you to see this, because this is going to help you understand just a little bit the importance. The first one's really important, and that's antibiotic resistance. All of these species over here, and many, many, many more, have picked up antibiotic resistance because they were transferred a plasmid that had genes on it that code for those antibiotic resistances. So that's a huge one that's really important to all of us, because that's happening all over the place. Another one we just talked about is the ability to make that sex pilus. E. coli, it's coded for on a plasmid. Pseudomonas, it's coded for on a plasmid. There's others as well that have the ability to make that. Even some gram-positives can make that sex pilus. This is an interesting one. There are some types of bacteria that cause disease in plants, and most of us like to eat. And whether you're a vegetarian or an omnivore, you still need plants. Because if you're an omnivore, the things you eat probably ate a plant, or else they ate something else that ate a plant. And so there's this guy that causes tumor in plants. It's called agrobacterium, and there's a number of species that they cause tumors in plants. The plants become unhealthy, and then they don't survive very well in most cases. And we'll talk a little bit more about that. The ability of rhizobium to fix nitrogen is also coded for on a plasmid. Rhizobium has been nominated as the state microbe of Texas. It's true. About 4 years ago, Wisconsin got the bright idea to have a state bacterium, and they decided to choose the Lactococcus since they make so many dairy products in Wisconsin. And so we in Texas, not to be outdone, said, wait a minute, we need a state bacteria as well. And it was about the time when all the oil was spilling into the Gulf, and so I suggested, well, the Vibrio fischeri, they're out in the Gulf, they can naturally break down that oil, they're actually saving a lot of it from getting to the coast, and we debated that, and it was a good idea, but we hope we never have another spill like that again, so I hope they don't persist, and they will persist, but not in the abundance that they were when the oil was seeping out. And others said the state flower of Texas is the bluebonnet. And the bluebonnets can't survive unless they have rhizobia at their roots, fixing nitrogen and feeding the nitrogen to the bluebonnet. So it was the logical choice for the bacteria, the state bacteria of Texas. It was sent to the state senate. I don't know if it ever got approved, but you can petition your state senator and tell them that they really need to make a decision on that. And so that's the rhizobia. They actually